Hello and welcome. My name is Colby Evans. I am a dermatologist in Austin, Texas in the United States, and the past board chair of the board of directors of the National Psoriasis Foundation. I'm here to speak to you today on biosimilars. We'll talk a bit about the history of biosimilars and how they are made, how they are approved, and then about some of the hopes and so far unrealized hopes of cost savings from these medications. They certainly are becoming an increasingly important part of the landscape of biological treatment across the world. These are my disclosures and potential conflicts of interest. You can see these companies that I speak and consult for. So on a basic level, biosimilars are a biologic medication that is highly similar to a reference product or a branded drug, notwithstanding minor differences in clinically inactive components. They should not be clinically any meaningful differences in terms of safety, purity, and potency. It doesn't mean there couldn't be, but there should not be for it to truly be biosimilar. And starting from public information and the originator biologic drug that's on the market, a biosimilar is created using reverse engineering to create a process from the ground up that can create a molecule that is very similar to the reference drug and then can be tested to prove that it works in similar. So we can see here on the upper left-hand slide an example of why biosimilars have generated such interest. If you look in the US at net drug spending, and of course the US is probably going to be higher for a number of reasons than much of the world, it is an increasing amount that is being spent on biologic medications. The blue bars here represent biologics. In 2013, about $77 billion. By 2017, that had gone up to $120 billion. And then, of course, over time, it has only gone up from there and is expected to go up further as biologics become first-line agents for more and more conditions. The top right and bottom left slide show the timeline of biosimilar approvals in the EU and in the United States. You can see there are many biosimilars that are approved in both jurisdictions, and there have been some that have been approved for many years and some that have only been recently approved, and the number is certainly increasing. The promise of all of this, really the only reason for a biosimilar, is to reduce cost and increase access to a biologic drug. If you've made your biosimilar correctly, there really shouldn't be a dramatic clinical or safety difference from the reference product. So the real purpose of biosimilars is to create competition in the market and hopefully decrease price and increase availability. When you develop a biosimilar, you do it quite differently than a traditional branded reference product drug. So on the left hand of this figure is reference drug development, where there's relatively less time and money and effort put into the chemical analysis and analytical analysis of the molecule, and then spending and effort increase as you move towards preclinical studies, phase 1 trial in healthy volunteers, and then ultimately your phase 2 and 3 studies, which will be large, often thousands of patients, multi-center trials, and can be quite expensive. On the flip side, when you create a biosimilar, you spend a lot of your time and money and effort in the laboratory taking the reference molecule and sort of dissecting it, learning how you can create it with your own processes in a way that will be not identical, but very similar to the reference drug. Then you can spend relatively less time and energy and money on preclinical work and confirmatory trials because of extrapolation, which we'll talk about in a minute. If you are able to confirm that the drug is clinically equivalent in your phase 3 trial, you may be able to acquire many other indications without having to do repeated large multi-center phase 3 trials.